introduction, UV/VIS spectroscopy is essentially technical for analytical thermal interaction in the ground of Escherichia coli, a key microorganism in biological and biotechnological research. Study how Escherichia coli responds to change in temperature, provide valuable information about its adaptation to different environment conditions, which is critical in food microbiology and medical research. This technique helps to understand how temperature affects the growth and adaptation of Escherichia coli. In this experiment, UV/VES stroke is used to analytical how different temperatures affecting the growth, rate, and generation time of Escherichia coli. UV/VES stroke in a current and widely used technique for measuring the density of microbial culture. Combining this technique with temperature variation provides the type information of how heat affecting the growth of Escherichia coli. These findings are critical to understand how the thermal changes can influence Escherichia coli proliferation, with important implications for food safety, disease control, and biotechnology.
In this section, we will know the general and specific objectives. The general objective is develop an adequate understanding of how different temperatures affect the generation time and growth rate of Escherichia coli, and is using UV and VIS spectroscopy techniques to measure microbial growth by performing precise calculations. For the specific objectives, we have the understands the proper use of equipment such as UV and VIS spectrophotometers, an incubator, as well as culture materials for the experiment. The other is grow Escherichia coli and expose it to various temperatures. The other is measure the growth of E. coli and analyze data and create graphs about the growth of E. coli and calculate the generation time. The materials used in this practice were Escherichia coli strain, suitable liquid culture medium, in this case natural broth, test tubes or eliminator flags, water bath or orbital incubator with temperature control, spectrophotometer, pipettes and tips, and vibrator.
I will present to you the procedure of the practice. In the procedure, first we must disinfect the area and keep our hands clean to use the laboratory instruments. Firstly, the E. coli strain was placed in 6 test tubes containing the appropriate liquid culture medium. 9 ml solution to 1 ml E. coli culture. A seventh test tube, which was used as a control, was designated blank and contained only the liquid culture medium without the presence of E. coli. Each of the tubes was labeled as follows: T0, T1, T2, T3, T4, and T5. Secondly, the culture was incubated at an optimal temperature of 42 degrees centigrade in a water bath to allow adaptation of the microorganism to the culture medium. Third, after preparing the samples, a spectrophotometric uh, study was carried out to determine the absorbance of each sample. Okay, to use the spectrophotometer, First, remove the sample in the correct weight using a burner and then place the samples in sterile cuvettes. It is important to handle these cuvettes carefully, holding them on the opaque side to avoid any contact with the surface where the light passes through. This is essential to ensure that no staining or contamination occurs in that area, um, which could affect the accuracy of the results obtained in the spectrophotometer. For the blind sample and T0 were prepared at 9:45 a.m. and measurements of both samples were taken using the spectrophotometer. From then on, the data of both samples were recorded in the spectrophotometer every hour. As a last step, samples were taken at 1 hour intervals: T1 at 10:45 am, T2 at 11:45 am, T3 at 12:45 p.m., T4 at 13:45 p.m., and finally T5 at 14:45 p.m.
Results. As was mentioned before, after making the incubation, we take the measurements every hour. This in order to know how the generation or growing the E. coli was going. So, in the table and in the graphic, we can see that at first it has a great growth value, but then it starts to fluctuate, increasing and decreasing as the time passes. Expected results. To give us an idea of how long it should take to our bacteria to generate, we can use the following formula, where the G is the generation time, T1 and T2 are the times selected, and N1 and N2 are the cells densities of in those times. To put into practice, we take the first two values, where the growth curve of the bacteria was best observed. So, as we can see, a result of 250 minutes was obtained, which means that it should take this amount of time to our bacteria to generate or to grow. Result obtained. If we take a look again to the table, we can realize that the value obtained to this experiment was 270 minutes. If we compare it to the expected value, which was 250 minutes, we can see that there is not a very significant difference. These 20 minutes may occur due to a slightly delayed data collection in each time interval.
Conclusions. The conclusions of this practice are based on the observance data of the 515 obtained at different times during the experiment. It is concluded that the cell density of E. coli increased as time elapsed. However, we did not observe a clear linear trend in microbial growth over time. In general, observance values varied, suggesting that the growth rate of E. coli was not constant over the study period. Significantly, the temperature to which the bacteria were exposed could have influenced this variability, as different temperatures can significantly affect the generation time and growth rate of E. coli. Recommendations for effectively carrying out this practice of studying thermal interactions in E. coli growth using UV-VIS spectroscopy are: calibrate the spectrophotometer using a target culture medium without bacteria, and ensure the measurement wavelength is correct, in this case 515 nanometer. As in this practice, it should be communicated if in the spectrophotometer has already been calibrated To avoid confusion, it is recommended to perform several repetitions as different temperatures, hygiene, and safety in the laboratory must be warranted. In addition, it is essential to consider the practical implications of the results and to comply with biosafety guidelines when handling E. coli.
And next, relationship between temperature and microbial growth. Question 1. How does temperature affect in the generation time of Escherichia coli? The temperature is a critical factor affecting generation time in Escherichia coli and most bacteria. A temperature influences the rate at which bacteria reproduce and grow. Bacteria have an optimal growth temperature. The temperature at which they reproduce fastest is between 37 and 42 degrees Celsius for Escherichia coli. This temperature is optimal because they have lower temperature. They reproduce show down significant, even stopping near freeze point. At At higher temperature, bacteria growth accelerates, but excessively high temperature above the 15 and 55 degrees Celsius can damage or kill the bacteria due to health stress. At extreme temperature, either be very cold or very hot, bacteria growth stop or slow dramatic. Question 2. What did you observe in the ground curves at different temperatures? How could you explain this observation? Temperature curves are used to visually and explain how temperature affecting the growth and activity of microbes such as Escherichia coli. I explain how to interpret and explain these curves. X-axis represents time, usually in hours or some other relevant time interval. This axis shows how the temperature and volume over the time. Y-axis represents a measure related to bacteria growth or activity, such as oxygen optical density or bacterial population. Optimal temperature when the temperature coincides with the ideal ground temperature for Escherichia coli around 37 42 degrees Celsius. Higher activity and growth is observed in the curves. Low temperature lead to lower lancity speeds, where bacterial activity is low and growth is slow. This, this is because Escherichia coli need more time to adapt and reproduce, leading to a delay in the growth phase. Elevated temperature accelerate the growth of Escherichia coli by reducing of generation time, leading to a faster growth rate. The temperature extreme very cool or very hot temperature lead to a triggers or restart of bacteria activity. Near the freeze point, bacteria activity stop. While at the high temperature, bacteria can be damaged, leading to a palliative or decline in the core. This curve represents how temperature influences the growth of Escherichia coli, with in place of dormancy splits, growth rate, and the impact of heat stress. They have application in microbiology, biotechnology, and release fields.
Question 3. Why is UVB spectroscopy used in this experiment. UV's spectroscopy used in this experiment because it allows the precise measurement of E. coli cell density as a function of light absorption at a specific wavelength 515 nanometer without destroying the samples. This technique is fast, easy to use, and highly reproducible, making it an effective tool to follow microbial growth in real time and understand how different temperatures influence it. Question 4. What wavelength is used to measurement absorbance and why? A wavelength of 515 nanometer is used to measure absorbance in this experiment because it is wavelength in the visible region of the electromagnetic spectrum that is within the sensitivity range of the UV vis spectrophotometer and is suitable for the detection of organic compounds that observe in this region. In addition, in this case of E. coli, the observance at 515 nanometer correlates well with cell density, allowing an accurate measurement of the concentrations of bacteria in the sample.
For the question 5, I explain how generation time G is calculated from observance data. First, I want to explain what generation time is. Generation time is basically the time it takes for a group of bacteria to multiply during their rapid growth phase. To understand this, we use a measurement called observance in an optical density at 550 nanometers, which gives us an idea of how many bacteria are in the sample. When bacteria reproduce, this observance measurement also increases, telling us that the bacterial population is growing. For the process, we have 5 steps, and the first is that select least 2 points or more in the exponential phase and choose two or more points in your observance data where you can clearly identify the exponential phase of growth. These points must be on the part of the curve where the population is growing exponentially. Second, uh, obtain the time and observance on optical density at the 500 nanometers values for the selected points, and we can record the time and observance values for each select point in the exponential phase. Third, calculate the observance radio. The observance radio must be calculated for each pair of select points, and 2 and divide n1. For 4, calculate the base 2 logarithm of the observance quotient. For that, we can take the base 2 logarithm of the observance quotient for each pair of points. And finally, with all the results ready, we calculate the generation time G. And for this, we use the formula you can see in this part. And this form, we can calculate the generation time from observance data. Question 6. How does generation time vary with temperatures? An important aspect to consider during the study of bacterial growth is that its behavior can vary according to changes in the standard conditions. According to temperatures, the standard condition is the ideal temperature of the coli bacteria is 37 degrees Celsius. This change in temperature can cause a decrease or increase in population's growth. We can see temperatures according the generation times E. coli. The optimum temperature are, we aforementioned around 37 degrees Celsius. This is the optimal temperature for the growth of E. coli. Under these conditions, its generation time is around approximately 20 minutes. For the case of low temperatures at colder temperatures, for example, around the 10 degrees Celsius, E. coli can still grow, but its generation times becomes much longer. And the case the high temperatures at warmer temperatures, for example, around 45 degrees Celsius, the enzymes in E. coli cells are damaged and the process is slowing their growth rate.
Question 7. What is the importance of understanding how the temperature affects microbial growth in the context of food microbiology and medicine? Well, growth time is very important in microbiology because it is responsible for studying the behavior of microorganisms as well as their main characteristics and their growing environments. So, with this in mind, it is more important in medical aspects since this can determine possible treatments to prevent the growth of these bacteria and thus also avoid possible diseases caused by them. Question 8. How could the results of this experiment can be applied in the food industry or biomedical research? Well, in the aspect of food industry, these results help us to know the temperatures at which these bacteria can be generated or grow exponentially, giving us a guideline for the treatment that should be given to the high-risk foods to prevent they contract these bacteria. And in the aspect of biomedicine, by knowing the behavior of this bacteria, we can go and search for a vaccine that will help in treatment to a person that could get infected by this one.
About this question, if the absorbance decreased drastically after reaching a peak, it could indicate that E. coli declining due to factors such as cell death from lack of nutrients, accumulation of waste products, or the action of antimicrobial agents. It could also uh, be caused by change in the physical environment, such as temperature or pH, or by the presence of competitors or predators that negatively affect in the bacterial population. When handling microbial cultures and equipment such as the spectrophotometer, precautions and safety measures are required to prevent cross-contamination, such as the first is wear protective equipment, that is gloves, safety goggles, and in some cases a face mask. Second is cleaning and disinfection. Uh, Wash hands before and after handling microbial cultures. Disinfect work surface before and after. Clean and disinfect instruments to be used. Uh, Third is cultures should be labeled with relevant information such as date, type of microorganism, and the person responsible. The last is adequate storage. Culture should be stored in appropriate conditions and at a specific temperature to avoid pollution. About the use to a spectrophotometer, It should be calibrated and checked for accurate measurements. It should be cleaned um, properly before and after use to avoid pollution, and samples should be handled carefully to avoid spillage and adherence to the surface of the equipment. Finally, proper maintenance should be carried out to avoid accidents and problems with the equipment. It's important to wear full laboratory clothing during an experiment for several reasons, such as personal protection, as it protects the experimenter from possible chemical splashes and prevents direct skin exposure to irritating or toxic substance. Other is prevention of cross-contamination. That is, proper clothing helps to prevent cross-contamination between different experiments or samples, environmental safety, um, minimize the risk of accidental release of dangerous substance into the environment as coats and gloves can trap spill or splashes, preventing them from propagating and causing further damage. And finally, compliance with safety protocols as the use of appropriate clothing is a mandatory requirement according to safety rules and regulations.